So without further ado, Professor Maritha Kotze is a human geneticist, and she's currently employed as a principal medical scientist in the Department of Pathology at Stellenbosch University and the National Health Laboratory Services. She started her career doing BSc, BSc Honours, and MSc, and passed all of these with cum laude. In 1990, she completed her PhD and thereafter supervised numerous PhD graduates, including me, as well as a number of intern medical scientists. Prof. Kotze has published more than 150 peer-reviewed publications, as well as been invited to several book chapters. She has received many awards, including a Patent Incentive Award for Research Innovation in 2005. Over the last 2 decades, Prof. Kotze has focused on research translation in her capacity as Director of Genomics, a spin-out company of the South African Medical Research Council. The pathology-supported genetic testing framework resulted from this work was acknowledged as best practice in 2020 by the International Consortium for Personalized Medicine. Tonight, we will hear a bird's eye view of how this framework can provide an approach that can effectively integrate research data towards clinical utility and to bridge projects from the study population to the patient or to the person. Prof. Kotze, I hand over to you. We look forward to hearing from you this evening.
Thank you for the introduction. It's really a pleasure for me to give this talk tonight. I've been listening to so many people over the past 2-3 years when we had this. And I was thinking, when will I now actually be ready to give this talk? And I think tonight is for me very special because we have received about 5 million Rand from the one fund, which I would like to acknowledge. Let's see. The Technology Innovation Agency. They really, in a way, entrusted us with funding to take the next step. We can do research, but to really translate it and to do it in a way that we hopefully could involve and engage many people across the different disciplines. That was something that is really needed. And I hope that we could make a small contribution as part of a South African scientific and clinical society. And you as one of my PhD students, Karen, I think the value that it brings for us to interact at the level that we did, this is what makes the whole difference. We can find that place where the knowledge that collectively is provided towards the benefit of patients. And I must always mention that the role of the genetic counselors absolutely necessary, and I hope that more doctors would make use of their knowledge on how to bridge those gaps. I think that is their role, is to bridge the gap, the genetic counselors, but I also realize that us as medical scientists must try and bring the knowledge that we have through the articles that we publish, through the students that put so much effort into each thesis. How do we bring it together? So the pathology-supported genetic testing is really what the names say. It is about having sort of a plan, a goal, and then everything that we do, all the research, every report that we write, how does it actually add to that value? So I shouldn't spend too much time on that, but hopefully you had time to read through this. So I do not have to mention specific people. There's so many people that contributed. So the students' names were mentioned. So breast cancer is one of the life-threatening, non-communicable diseases that really, is the forerunner in personalized medicine or precision medicine. Now the pathology-supported genomics, the whole idea is to screen for multiple NCD pathways that would be relevant to an individual, and then perform selective genetic testing. The title of the talk is also focusing on postmenopausal breast cancer as the comorbidities becomes more important. the familial risk may be less important in the way that we look at age as one of the selection criteria usually, and then pharmacogenomics goes across both. But what I want to try and convey tonight is that actually for all breast cancer patients, all three of those factors are very important. So the Open Genome Project's website is shown here, and it's really about bringing about how your lifestyle, your family risk, and the therapy can all be bring together into the treatment domain where we as medical scientists hopefully can then also make a contribution. So as breast cancer is definitely not one disease, it is important just to show a picture like this to show us that all these different aspects are important. The histopathology, where we mostly the most common forms are the invasive lobular carcinoma and the infiltrating ductal that has now been classified as carcinoma of no special type, the new name. And then all the immunohistochemistry and for estrogen and progesterone receptors, and then the HER2 where the fluorescence in situ hybridization is added if there's equivocal. So it's very important for us as scientists then to also have learned the pathology terminology to be able to take the genetics through the pathology and place it into the clinical treatment domain. So this is the whole idea with the pathology, to make that bridge. So the problem is really that large numbers of patient samples are currently leaving African countries from both private and public institutions for genetic or genomic testing. And there's nothing wrong with it if we look at the turnaround time, the interpretation challenges, and high cost of care. If we cannot do it in South Africa, the patient still comes first. But what we must always strive for is to bring those technologies into South Africa. So this is part of this project, and I think we have come a far way. So a framework for the implementation of genomic medicine for public health in Africa has been published in February 2021. I was part of 38, I think, African scientists across the continent, and this picture that's shown on the right-hand side was part of that. So because we realized that the solutions would require collaboration across boundaries. And this is something that I always like to do. I really like to interact with people when it comes to the science. And I just want to read this figure legend. You know, sometimes it takes you a long time to try and find the right wording. And I'm just this person that would read back my own articles many times because there's so much care in a way that goes into that. And I think if we read this, it will make it clear that it is possible to use a three-pronged approach. This wording was also coined in a way during the PhD study of Prof. Karen Baekes. So a pathology-supported genetic testing framework for application of three-pronged approach towards identification of firstly then pathogenic mutations associated with familial risk. Then secondly, genetic underpinnings of tumor subtypes, biochemical abnormalities or comorbidities influenced by modifiable environmental risk factors. And then third, patients at increased risk of medication side effects or failure due to genetic variation. I think over the years, I realized that those are the three major things when we look at multifactorial diseases where there is a genetic component, where all those aspects should be analyzed. And at the moment, they are still very much fragmented. And I think this is also showing the picture of Karen here, was the whole purpose also of her studies. If we focus then on postmenopausal breast cancer versus premenopausal instead of maybe age, already just talking about post and premenopausal already bring that cancer into the treatment domain because the therapy would be different in those two groups, just starting with that. Okay, so again, just to emphasize, because I think many times, we use these words and do we really know what they mean? It's a dynamic multi-assay platform to address different aspects of the same disease in a patient. So it's like this where we say you don't treat the cancer, you treat the patient. And then to bridge the implementation gap through pathology, to bring the genomics into the treatment domain for clinical decision making. So Prof. Baatjies then published this article as part of a PhD in 2019 in PLOS, where she showed that postmenopausal breast cancer patients treated with aromatase inhibitors at Tygerberg Hospital, the high risk of bone fractures. About 1/3 of them fulfilled the international criteria for bone protection pharmacological intervention. And then there was only 7% who had sufficient vitamin D levels. So that is why the, sort of highlighting the differences in the histopathology of the breast was important, because in the forthcoming slides we would focus a bit on the lobular breast carcinomas. So if we look at the right-hand side, Professor Baatjies developed a database that other students could subsequently use in their studies. So from her clinical knowledge as a surgeon, she then opened the path for other students to use the same database and always been involved in those studies and helping us to not misinterpret the clinical information. So the entry criteria basically was that these patients were eligible for aromatase inhibitors. Then we looked at the research was performed at the interface between the research and service delivery. The clinical profile at baseline, which was routine, is shown here on the left-hand side. Age, personal and family medical history taken. Some of these patients received the genetic counseling as the standard of care at Tygerberg Hospital. I think we had about 8 patients in our group of around 100. I'll show you the numbers later. That had the genetic counseling and was sent for BRCA1 and 2 testing. And I think there was 1 patient with a VUS, had a more extended testing. Then the BMI was always taken. And then on the right-hand side, we included then as part of the research questionnaire more lifestyle factors and family and personal history of fracture, and then the oral steroid use. Then there were some special investigations specifically paid for through the research grant. Bone mineral density scans were done, and then calcium and vitamin D status, especially the vitamin D status is not routinely done in patients, and there some patients would get calcium assessment. Then the clinical intervention, what the doctors would then recommend based on the outcome and the results is exercise recommendations, just talking that through with the patient, the need for supplementation. Then some of these patients, this is where change of treatment comes in. So in this study, there was already some patients that because of the scans that was done for the research, they could not proceed with the aromatase inhibitors and were changed to tamoxifen because of the risk of the aromatase inhibitors for bone loss. And then consider also the biphosphonate treatment. And on the other hand side, we got ethics approval for patients to also sign my consent form, where the whole exome sequencing was then indicated for them to say yes or no. We also added a pathway-based analysis of more low penetrance genes that could interact or together with the environmental factors and the lifestyle factors that we documented in the questionnaire may increase some risk factors. And then the comorbidities, we kept in mind the aromatase inhibitor side effects. So people with joint pains, like the questions was asked about that, or whether they already had maybe been diagnosed with osteoporosis or bone fractures. Then also the breast cancer genes, I think at the time we screened 29 genes for familial risk in the study. Then the whole exome sequencing report was only provided to a small selection of patients, again from the study population to the person, only after genetic counseling. Then on the service component, the genetic counseling stood out as a very important aspect of genetics, and then screening for pathogenic mutations in family members as appropriate. And on the research side, we could then build a database for health outcome studies or further studies. And in the process, we then developed an adaptable report template that still gets improved continuously. So why screening for vitamin D deficiency? There's a lot of controversy in the literature. Vitamin D deficiency is not important, but it is undisputed in the prevention of osteoporosis and bone fractures. We really think it is important, and currently it's only recommended to test for vitamin D deficiency in high-risk individuals, which include patients with osteoporosis, patients with a malabsorption syndrome, obese persons, and patients with disorders that affect metabolism of vitamin D. There's modifiable and non-modifiable risk factors, and very important to remember that vitamin D exerts its action through the vitamin D receptor, and the vitamin D receptor can activate many, many genes, and it even play a role in DNA repair. Then we feel that, studies have shown that the relationship with breast cancer is subtype specific, and just refer back to the initial slide, the importance that breast cancer is not one disease. So someone do a study and they group all types of breast cancer together, you will not be able to find any associations. So what this study from Kim et al. In 2011 showed is that vitamin D deficiency correlates with the poor outcomes in patients with luminal A and luminal B breast cancer. And then studies also suggest that the active form of vitamin D protects against breast cancer by activating the vitamin D receptor. So what we were focused on is that osteoporosis is a major comorbidity in hormone receptor positive breast cancer patients treated with aromatase inhibitors. So the focus is here on the postmenopausal patients, because they have replaced tamoxifen as the first-line treatment. And this basically happened at the entry of, at the beginning of Prof. Baeke's PhD. So this whole process that we walked through. So the next student that then used the database of Prof. Baeke's was Dr. Abisola Okunula. This comes from his PhD thesis, just to show how the patients were selected. Again, we can just see that the pre and the perimenopausal patients were excluded. We selected the patients all on vitamin D levels if we had enough, if it was available in the database. And then there was also 3 study groups, and this is why this study of Abisola was so significant for me. And because in the same study we could address lifestyle risk factors where vitamin D comes in, the bone mineral density scans, even without genetic testing. A change of treatment could be implemented, and there's no time today, but Prof. Baeke's also published a paper on the CYP19A1 gene with some single nucleotide polymorphisms that we looked at and selected one beforehand based on the evidence in the literature, which then proved to be very important in the study population, because the majority of patients had insufficient or deficient vitamin D levels. It is possible that the effect that we saw with the CYP19A1 gene would be expressed against that background, but we are busy with further studies around that. And then obviously the tumor histopathology. We know that triple negative breast cancer differs from hormone receptor positive breast cancer. So familial risk would also impact on, so the genetics could also impact on the tumor histopathology, which we will show a bit later. Then here is just showing that from the study of Abisola, it could show that when the vitamin D levels were determined during the winter, it was significantly lower compared to the summer. And then also there was a significant association with the body mass index. Now we all know that from the previous slide that a high BMI is a high risk for low vitamin D. And we felt that this results that is shown here on the left-hand side was actually very important for us to give the confidence that that the vitamin D levels that was determined for his study and initially with Karen was we can trust the results. So it's a case of validating in your own population these effects. So I just wanted to throw in this slide on the right-hand side, because when us as scientists, when we are busy with one study, our minds already linger to going to what's next. So I'm very happy to also tell you that we got a very significant grant to look at COVID-19. There's also a PhD student that would be looking further into that, and the VDR and DSNPs that Abisola has now standardized in our laboratory that I will show in the next slides. another student from a clinical side that is not doing herself the genetic testing at the moment can now benefit from his study. So here we have used, in our study population of just above 100, there was only about 5 people with sufficient vitamin D levels, and they are shown here in green. You can see above 30, 39. So these levels were above 30. Then there was about 5 with 12 or less, which is very low levels for vitamin D. And on the right-hand side, I'm just showing the vitamin D receptor binding protein, how that binds the vitamin D, and then it interacts and activates the vitamin D receptor. So it's like a knock-on effect that we must understand. So when we look at the first one as an example, we can see that this patient with the ultra low levels, the blood was taken in the winter, the patient was obese, she was a compound heterozygote for 2 SNPs. And when we extended that SNP analysis to about 100 patients in his study, we could not show the gene effect on its own. And this is what was for me such so important finding, because we know that it's not always the genetics. And when we tried to publish the paper separately on this data, the reviewers came back to say, oh, but we can't publish it because the genetic results were not significant. And they missed the point. The point is that we risk stratify. So we want to try and identify through, yes, using genetic testing. Is it a genetic, is genetics more the risk associated factor, or is it more the environmental factor? And in these patients, it was quite clear, even before we looked at the genetics, that the environmental factors are very important. So then this is just a very busy slide showing then when we moved on to the vitamin D receptor, I've just explained previously that the vitamin D levels can activate the vitamin D receptor. And now we must also remember that if there is a variant in the vitamin D receptor itself, the effect can be the same. So we can have vitamin D that's too low because of maybe environmental factors or interaction with low penetrance genes. I see it more on the environmental risk, lifestyle risk side. And then we could have genetic variation in the vitamin D receptor gene itself, and that can then make that the vitamin D receptor doesn't work optimally. So we could see then when we looked at well-known functional SNPs in the VDR that Abisola then found these significant findings in our study population. So you can also see then that the vitamin D levels itself, with the area under the curve, it could now distinguish between people with differences in their bone mineral density, while the genetic testing could do that. This is just a quick slide to show that we looked at tumor histopathology, and then if we look at those with the sufficient values, none of the patients had the lobular, invasive lobular carcinoma. So I got very interested in looking further at the CDH1 gene, because we know that loss of the Gknowmix expression due to a mutation in the pathogenic mutation in the CDH1 gene is a characteristic feature of this invasive lobular carcinoma. So Abisola then had to go back to the ethics committee and apply for further ethics approval that we could screen all the patients with where we have already whole exome sequencing done, or add more patients so that we could look at this gene, which he then was granted by the HAREC. So this is just a slide again. Now we're coming back to the familial risk, because with the CDH1 gene, he used the Minion long-range PCR device. It's like a hand-sized device, and he compared the results with the whole exome sequencing results, and we could show that the results were the same. So we didn't find extra mutations. There's always the question about maybe low coverage, uneven coverage with whole exome sequencing, but according to his results, that was not a problem for the CDH1 gene. You could also then see that sample number 19, 38, and 105, although it was hormone receptor positive patients and postmenopausal and those at risk for a BRCA mutation, it's already been tested and excluded from this analysis. we still found BRCA mutations. And then there was also another BRCA2 mutation that Prof. Baekjes found in the subgroup of patients that she analyzed, where there was more than 5% bone loss after the aromatase inhibitor treatment. So there's about, we did waste in about 50%, 50 of these patients. So we got a 10% in this hormone receptor positive group, and it wasn't even in these patients the founder mutation. So it just shows us that it would really be important to move towards a more comprehensive screening, where we may start maybe with a small test, a founder test, and then immediately move on to the NGS should that be negative. And if we find it positive, then it is actionable.
No więc właśnie ona powinna to zrobić. To już
Then I would also like to say that
Professor Kotze, if I could just interrupt you for a moment. Colleagues in the background, if I can please ask if you can mute while Professor Kotze is speaking. We can hear some speaking in the background. If all colleagues could please at this moment mute so that we can give our attention to the presenter.
So
I do not want to name colleagues that remain unmuted because we can see who's unmuted. Please, if you could just mute.
So there was then also individuals where we found a BRCA1 founder, a BRCA1 pathogenic mutation together with a rare CDH1 variant. So the question was, what is the classification? So again, here the histopathology came to our rescue. We could look at that. It's currently not included in the classification systems that's used in most laboratories, but we think it's very important. So in the research setting, which is the study, we would say we should consider breast cancer histopathology and variant classification tools to facilitate the interpretation of sequence variants, as evidenced by our findings. So there was a CDH1 variant at codon 224 that was detected in a patient with the ductal carcinoma classified as benign, based on the knowledge that pathogenic germline variants in CDH1 is exclusively associated with the invasive lobular breast cancer when it's germline. And then on the other hand, we identified at codon 433 also an invasive lobular carcinoma with this pathogenic BRCA1 mutation. And it has been classified as benign in ClinVar. But now we are wondering if it maybe have a modifier gene effect, because that is because of the fact that BRCA1 is usually associated with triple negative cancer, and we're now working with hormone receptor positive cancers. And also this family had gastric cancer. We also know that diffuse gastric cancer, the CDH1 gene is also very important there with germline mutations. Also then in the service setting, we detected both a pathogenic CDH1 variant and a likely pathogenic CHEK2 variant in a patient with a mixed ILC and ICNST tumor. Then this is just to come back to how we can now integrate these different pieces of information into the multi-assay platform. The value that was added through our BISOLA study in relation to postmenopausal hormone receptor positive patients relating to the diagnosis. What is causing this disease? Is it lifestyle factors? Is it inherited? We could see there was quite a few patients where we could find pathogenic mutations. So it could help to diagnose inherited cause of the cancer. These patients were all at risk of osteoporosis as a comorbidity because of the aromatase inhibitor therapy, but then we also identified 14 patients at baseline who already was diagnosed on the scans with osteoporosis. So most of them, depending on the level and many other factors that the doctors took into account, were then excluded from the aromatase inhibitors. And then, so we would then say the value that was added is the lifestyle triggered vitamin D deficiency, the obesity and lack of outdoor physical activity is important there. For the genetic predisposition to osteoporosis, we could see that the VDR gene plays an important role in these patients already at increased risk of AE-induced bone loss. And we could then see also that the genetic variations associated with osteoporosis at baseline, and then also bringing in the study from Karen that I referred to earlier the CYP19A1 gene, it was different variants that could cause the RE-induced bone loss versus the causal factors for osteoporosis at baseline. And then the tumor histopathology and familial risk. So the clinical relevance of the pathogenic BRCA1/2 mutations should then be interpreted in relation to other high-risk genes like CDH1. So if we then screen family members and we have patients in the family with cancer but without maybe the one mutation that was identified as causal, it just shows us to, if we look at the tumor pathology, it might bring more information on what to do further. Do not need to go much into this slide. It's just to say that since we can add the research component, we can now look further and study further this effect that we see looking at the tumor pathology and the genetics all together. And I really feel that clinical pathology, which is now the vitamin D, is really an underutilized phenotype for us to look at cumulative risk that can be caused by a combination of genetic, lifestyle, and other environmental factors, so that we can then target them for monitoring, and when there's treatment failure, that would inform then further investigation. This is just to show return of research results. How can we do this while we do research? I'll show you in the next slide. One of our postdocs from Kenya has actually illustrated how the process worked in her environment. But what Abisola has provided in his thesis, again, is how from his study population, he could move to one person. There was one person that Mardel Skuman at Tygerberg has actually counseled based on BRCA mutation. And she could, the test was, new blood sample was drawn. The sample was sent to Bloemfontein, where the BRCA testing is done. Prof. Dr. Nerina van der Merwe and Jaco Oosthuizen, they tested it and confirmed the West result. So this is just the process that we then use. And we can also see it here in the framework that the RISPA published in 2020. Just the most important thing is that there is now this duty for medical scientists like myself, who is registered with the HPCSA, to return and reclassify and reinterpret results. So if our informed consent forms is not allowing for that, it would be very difficult. So the database is important. So our informed consent do allow. And then we look at the database, because if we do not have the relevant data, we cannot interpret the results properly. And then we can just see again here on the right-hand side that the pathology-supported genetic testing facilitates the evaluation of all these clinical characteristics of each patient in relation to their inherited lifestyle or therapy-induced risk implications. So this is basically my last slide. I want to congratulate Lwando Mampunye. He wrote this article on pioneering BRCA1 and 2 point-of-care testing for integration of germline and tumor genetics in breast cancer management. A vision for the future of translational pharmacogenomics. Just to make it clear that we will not do a point-of-care assay and think this is now the answer. This is to, you've all seen the complexity that we had to go through. It is really to show that we can simplify things. We can use a test like that as an entry level. And then once that person is in the pathology supported framework, in many other aspects that we now refer to could be evaluated, whether it's relevant or not for each individual patient. So just to start the process, we realized that if we could have a test like this, it would be very valuable. We even did a survey. And what came out of that is if we can combine the point-of-care assay with a genetic counseling service, it's not necessarily that the counselor need to do the test themselves. They could work very closely with a technician or someone, but that would be seen as one action to increase the access of the testing in South Africa. Then in this study, Lwando validated 8 common variants, found a variance in our population and recurrent variants. But then came along Jaco Oosthuizen from Bloemfontein that's been doing this research for many years. And he identified another founder mutation that we also published together. And he will now extend this assay adding the new founder variant and then redo the whole validation process as part of his PhD. So Luando also won an Innovation Hub award of about 100,000. So I'm very proud of the students. And Jaco also is working with Nerina van der Merwe, and they developed the NGS panel for the NHLS that is focused on the DNA repair pathway. So it includes BRCA1, BRCA2, and many of the other genes that is involved in that pathway. There's a 15-gene oncomine assay. They've done about 400 patients so far in the NHLS. And this is also to simplify the process to say, okay, we've got a point of care assay. Next, we have the gene panel assay. And if patient still is uninformative, we can proceed with whole exome sequencing and bring that data back into building new assays, improving the assays all the time. Then I just want to step 10 years, 20 years back. So in 2010, I was invited to a conference in Egypt. And if you read actually this, look, you can see it's very ancient, but it would say that this idea can be taken forward by any number of participating scientists, genetic counselors, and clinicians without limit. And I'm so happy to say that really, I think this is happening. The future is really bright for us, and if we grow together, it can be amazing. And we've just also developed an app. It's a free app. University funding contributed to that, and I already consulted with 2 doctors, one from the private practice and one from the public hospital, to help us with the information. The app is already available on a web base, so just go gknowmix.app. And you can test it out. You have to be registered as already in the website. It's only for healthcare practitioners. So at that note, I would like to say thank you for your time.